Hi, everyone. I'm Salma Quraishi. Welcome to Neuroscientists Talk Shop, the University of Texas San Antonio's neuroscience research podcast. So today is February 4th, and we're talking with Zane Lybrand, who has just started as assistant professor at Texas Women's University in Denton. Hi, Zane.
Hi.
Hi. So until very recently, Zane used to be one of our own here at UTSA Neuroscience as an integral part of Jenny Hsieh's research group. His work there had a few different strains that concerned IP stem cell models of disease, and then also mechanisms of epileptogenesis. And that's what we're going to talk about today. So for his own research program at TWU, he'll be taking these tools in tech and stem cell bio to work out some of the basic science of regenerative medicine therapeutics to treat TBI. Do you want to say something about that, Zane?
Sure. So we'll kind of get into it today, but my background is really neuroscience. And I joined Jenny's lab a number of years ago and kind of became a stem cell biologist. And my independent direction, my new lab, really is kind of an intersection of the two. So we focused for a long time, especially this paper we'll talk about today, on epilepsy and mechanisms of epilepsy, and it kind of came out as a tangent. It wasn't really what I initially set off to do. So I'm excited I'm able to kind of get back to that initial question that kind of got me interested in science in the first place, and that's brain regeneration and why the brain itself doesn't regenerate, why it can't repair itself. And so we're using stem cells to model different traumatic brain injuries. So this is growing organoids, cerebral organoids from different stem cell types, embryonic stem cells and induced pluripotent stem cells, and really trying to dissect out the neurobiology of non-traditional brain trauma. So severe brain injuries, impact brain injuries, are pretty well studied. Animal models, rodent models, pig models, cell models, humans, people have really studied this for a long time. We're really starting to appreciate how much we don't know about non-traditional forms of brain injury. An example of this is like a mild repetitive brain injury, something that may or may not, give you a concussion, but repetitive incidences over time, we know are starting to lead to possible neurodegeneration, chronic traumatic encephalopathy, post-traumatic stress disorder. So we're using these stem cell models to try to dissect out these underlying mechanisms in the neurobiology of this. At the same time, we're trying to understand therapeutic ways to help repair the brain. So if you've got a severe brain injury, right, the brain will not repair itself very well. But are there things we can do to encourage it to repair itself? Can we help it repair itself? Can we build, things that can help promote the regrowth and the regeneration of these brains?
Super. And so you're just getting started this month in a beautiful new lab and recruiting students. So good luck with that. We hope to hear more from you independently. So we're really lucky today to have Jenny Hsieh. Hi, Jenny.
Hi, everyone.
And we also have first time joining us is Chris Navara, who is our associate professor here at UTSA and also our resident stem cell maven, huge history in stem cells, which we should do a podcast with you at some point to talk about some of that. Okay, great. So let's get started. So I'd like to take today to allow you guys to preview for the audience some fantastic new work that's literally in press as of today, or you just found out today. I'm not sure when it'll be available. So, and that's at Nature Communications. So in it, you painstakingly connect the dots between seizure insult and aberrant maturation of adult-born granule cells, and then the generation of spontaneous seizures. It's all sort of the hallmark of epileptogenesis. So you report a mechanism that's based on aberrant calcium dynamics in these immature granule cells, and that you found is GABA-A dependent, and that it occurs in a very well-defined time window. So the study has some really interesting and heroic technical feats that point to causality, and of course, some really fundamental implications for therapeutics in terms of disrupting epileptogenesis. So I hope we can talk about both, but can you first just talk to us about introduce us to the study, and especially about some of the fundamental questions that you were trying to get at with it?
Sure, I think, so in the field of epilepsy, there's this really strong pull to start developing true anti-epileptic drugs. There's a lot of medication right now. There's a lot of pharmacological therapies for patients with epilepsy, but they really function to suppress seizures. They don't actually get at the underlying cause of the disease, which it's complicated in its understanding because there's a lot of different ways epilepsy can come about. And there's a lot of push to start developing therapies that, are more preventing the development of epilepsy itself, this epileptogenesis. And we really kind of approach this as, can we start understanding epileptogenesis, the generation of this disease? And it became very apparent that these adult-born granule cells, these resident stem cells in the hippocampus that are continuously giving rise to new cells, offered an opportunity to do so. Right. So this paper piggybacks off of a previous Nature Communications paper where we basically identified these aberrant new neurons as pro-epileptic neurons, and we really just trying to understand a mechanism, like what's forcing them into this abnormal development versus a normal development. And I think that's really the heart of it, is we think this aberrant neurogenesis offers therapeutically a targeting that we could use for a true anti-epileptic and not just an anti-seizure drug. But if we can understand perhaps the molecular mechanisms behind it, then we can start to underlying disease genesis.
So when Zane came to the lab first as a postdoc, I think he was hoping that he could study the role of these adult-born granule cells and learn that if he understands the mechanism, he can learn how to promote regeneration. But after many years of working on this, all of the conclusions, all of the results sort of point to that there is this darker side of adult neurogenesis. And I think there's a lot of implications as well for when you're trying to replace neurons in the brain, either through exogenous stem cells or exogenous neurons. And Chris can tell us also about sort of his thoughts in that area.
Yeah, sure. I think that it's interesting to think about whether this is a problem of the stem cells themselves, or it's a problem of timing in the stem cells. And so I think it is very interesting to think about not only how you replace stem cells or repair with stem cells, but how you control for incorrect stem cell function.
So one of the hallmarks of these aberrantly or these newly born adult stems or granule cells, sorry, is their mismigration. I think that's sort of the one benchmark thing, and that leads to all these sort of network miswiring and recurrency, and you've really borne some of that out. But some of the really fundamental stuff is like, how do we, so can you just say something about the timeline? Because the time is very important in these studies. So there's an initial seizure or pilocarpine insult, and then there's a period that after which all the neurogenesis that happens then ends up somehow modified. And so what you're looking at is really that early time window. Is that right? Can you just say something about the actual technical part about the work so that we can?
Yeah, so, excuse me. So the original Hsieh et al. 2015 paper, we used a transgenic approach to ablate the cells. And one of the caveats with that is once we've ablated, they're gone. We can't study them anymore. So we can't study why anything's happening to them. So we wanted to switch to something that's more modulatory, something we can turn on but doesn't remove the cell, something we can turn off, but this, we can still study the cell, right? So we switched to optogenetics, we switched to DREADS, mostly because we could control the timing of all of these, because we really wanted to focus on this early time point. So in these rodent models, you give a convulsant, pilocarpine in this case. There's other ways to do this too, with canic acid or something. And then you wait a period of time, and during that period of time, this latent period, it can be a couple weeks to a month. That's when the seizures progress. That's when you have these circuit changes. That's when you have the cells dying. That's when you have basically the brain permanently changing so that you develop the epilepsy. Well, what's nice is we can also coincide the maturation of the stem cells with that same time. So it's almost like they line up together. So cells that are born right at the time of this injury, this pilocarpine induced injury, the timing of their maturation, it's about 4 weeks, about a month or so, matches pretty closely with that latent period of seizure development. So to us, that was something that was pretty strongly correlative in terms of studying. So we're trying to also piggyback on terms of that, right? So then for us, basically the incentive of this was, it's got to be an early period, right? If these cells are born right at the time of the injury, there's got to be something immediately disrupting the way that they develop. This comes from a lot of former work from Jack Parrott. So he mapped out a lot of how these cells evolve. So if you label old cells, young cells, new cells at all ages, he really showed that it's the most immature, most actively dividing stem cells that give rise to the bad cells. If you look at the mature cells at the time of the seizure, when they're fully mature, they don't really change that much. They may change a little bit in response to the seizure, but there's no permanent alterations. So it's really this opportunity for the stem cells when they're essentially born into this trauma environment that forces them to develop inappropriately.
And one of the hallmarks of that is this increased calcium that you are then able to modulate and really just basically reverse the course.
Yeah,
or you think reverse.
Yeah, so there's a whole list of things, a whole list that people have identified in terms of what these cells do. And part of the struggle we've had is dissecting them out. So we've got this ectopic migration, we've got these mossy fiber sproutings, we've got hyalobasal dendrites, and there's some camps that are really favoring one versus the rest. But we're still struggling to dissect out to see which one's doing the most or contributing the most to something, right? So at this point, we still haven't.
Or how related they are,
how
right?
related they
because
are,
it seems it seems like when you reverse calcium, you change the way the dendritic migration happens, right?
That's right. So I think the nicest, the cleanest things that we demonstrate in this paper is simply by activating calcium with this HM3DQ dread, we cleanly see there's a mismigration, and that's associated with seizures, right? We don't really see any of these other cell changes. And to me, that's a really clean way to understand, uncouple some of these features that we haven't really been able to do before. And so I think we're kind of starting to get there. And so I think hopefully moving forward in the future, this is giving us an approach to do so, right? So is this calcium? What can the calcium be doing to migration cues, right? So then downstream of calcium, are we disrupting,
It's super compelling because the structure function relationship of the network is actually, it's completely laid out in this story, and you get more recurrency, you get less cortical integration happening. I mean, it seems like from start to finish, from calcium all the way to the network, that it's just like it's a sort of a great roadmap to start thinking about. I mean, super exciting, great work.
Thank you. Yeah, I think it really, we took it step by step. Why was this happening? Somebody asked us, why is this, when you do this, why would this happen? Well, you should look at this. So we look at that, and then that would lead to the next step. We're like, well, if we're changing the position of these cells, if we're forcing them to migrate differently, we're clearly going to change the circuit, right? I mean, I think that's inevitable. So how is that doing? How do we do that? Well, what can we do to approach that? How can we answer that? How can we monitor that? So,
Zane, do you have thoughts on the persistence of this? I mean, I think it's easy to understand that an insult might cause a transient problem, but this is a problem that persists. And so why don't the cells just fix themselves down the road after the insult?
That's a good question.
So I think, one curiosity is how generalizable is this to other insult models, injury models, or even epilepsy models? And so there's another, maybe Zane mentioned it, but one of the leading causes of epilepsy is traumatic brain injury. At least us and other groups, attempting to model post-traumatic seizures, not all of the mice develop epilepsy, even though we very, try to carefully control the initial insult. So I think the question is why are there these differences in the disruption of the circuit, the formation of epilepsy between different animals who are otherwise, similar genetics, similar injury environment, and so that's a very important question that I'm not sure we're interested in addressing, but I'm not sure at this point we really know. And if it has to do with this calcium at this early period.
Yeah, so I think, a lot of it also comes down to uncoupling these characteristics that we can identify, the migration, the dendritic changes. And I think different trauma events, traumatic brain injury, pilocarpine, they change a bunch of different things different. And we know, for example, there's, it's published where if you take a DAB1 knockout, which is, critical and renin signaling, so that it's a cell migration cue, you can drive ectopic granule cells, but the mice don't develop seizures, right? So we know there are models where it's not sufficient, that it's this migration alone to cause seizures. So then there's got to be other things involved, right? If that's, the pilocarpine model itself has got a lot of issues. It's not a clean model. We're changing a lot of things, right? Traumatic brain injury, if it's generalizable to other trauma-induced epilepsies, traumatic brain injury changes the hippocampus tremendously, right? It completely transforms it if you look at these models completely. So I think we're starting now to try to uncouple a lot of these things that might start to answer that question, Chris, about like what's causing this persistent. And I think it gets down to that not all of these aberrant neurons are the same. Some of them are more aberrant than the others. Some of them are possibly still good. Some of them are, more bad, I guess you can say. And I think it comes down to that, and I think we're going to find out there's just heterogeneity in these populations that are going to inherently make it more difficult to understand.
One of the really interesting things about this is the mysterious nature of any neuron during maturation, right? There are all of these dogmas that we have about cellular cascades and about like GABA hyperpolarizing versus depolarizing, right? That we take for granted, but it's kind of and calcium is such a ubiquitous signal, and you can imagine that at different time points in maturation that a calcium load is going to have a completely different explosive set of effects on, gene regulation, on all kinds of structural things. So it's kind of interesting how the migration is so stereotyped, or is it that stereotyped, or is it just all over the place? Because you would imagine that there would be a lot more variability, or maybe there is variability. Is there variability in the migration? It doesn't go where it should, right? It's not all sort of in one direction or another. Is that right? Or
it does somewhat, and so if you compare like a normal mouse versus a pilo mouse, it kind of disperses both directions, but mostly, inappropriately into the hilus, which is that's where we define these ectopic cells. I would highlight that we're not focusing on, like, this paper doesn't also just focus on these cells. It's very technically difficult to just focus on these cells. I think we all kind of want to, but we can't exclude that a lot of the analysis, you might have aberrant cells that look normal, right? So we can't really exclude how, just the way it looks, what it does. So if you patch all of these cells, some of them in the hilus, they're functionally different than if you patch a cell in the subgranular zone. Well, if you patch a separate cell in the subgranular zone, it may still be different, right? It may be a third type of cell. So there's such a variety in the way that these cells develop that I think that's what's really difficult to say. And it could be that that's what calcium does. So for the ectopically migrating cells, maybe calcium response is more exaggerated, right? So in the paper, I basically plot every cell in the analysis, and you can see the diversity in the response. Some of them it's crazy, and some of it looks kind of normal, right? It could be that those crazy cells are the ones that will migrate ectopically, right? And then some of them just don't. So we haven't been able to do any kind of like lineage tracing and understanding that. This was the first time we've started looking at it, so I think it could come down to something like that, right? Should we just have different responses in these cells that might drive different developmental programs, whether it's gene regulation or membrane properties? It's difficult to say at this point.
Yeah, I think that's a really important thing to not leave people with the impression that we know what makes what really is different between aberrant and non-aberrant cells, and we don't think it's strictly due to location. In fact, what Zane was saying, a lot of the analysis of the cells was done unbiased and not preferring one location over the other. But it would be interesting to eventually identify what are there genes that are specifically activated in aberrant cells, and if we can find those specific genes, we can maybe use them, use the promoters of those genes to then go back to make these tools even more specific. But at this point, we don't have, the tools themselves are not as specific as we would like.
So something else that you guys have written and are obviously thinking a lot about is the genetic diversity of epilepsy models, and how in vivo models like the ones that exist, and of course you mentioned pilocarpine is not the only model that people use in vivo. So how IP stem cells are really going to help get at some of the kind of issues in there that are embedded in some of that genetic diversity of things that cause epileptiform activity. So can you say something about what the general mechanisms that you're working out in this sort of study, and how you're sort of going to take those and run with those in this other model, which has another level of complexity to it? Like how is one thing going to inform the other, or are these just two completely different sets of explorations that are going to hopefully land in some common areas? No one wants to touch that.
Jenny, you can touch it. I can.
Zane, you can start.
We can just move on to the next question.
So no, it's a huge question. So we had a neurologist, pediatric neurologist, join the lab years ago, and I remember in the lab meeting we're talking about pilocarpine, and he was like, "Why are you guys studying acquired epilepsy? Like 70% of patients with epilepsy, it's not acquired; it's a genetic epilepsy. So why are you focused on the 30%? Right? Why are you spending resources and 8 years of your life trying to understand something that's affecting 30% of all the people we treat, right?" And it really opened my eyes, because then I was like, yeah, why am I doing this? But I think, to get back to what Jenny was saying, generalizing this to other models, I think that's still a huge question that we have to see. Do we see aberrant neurogenesis in patients, right? We can get resected hippocampus tissue, we can stain for some of these markers, and we can start to see a lot of the adult neurogenesis in humans that's being published comes from resected epilepsy patients. So it would be really interesting to see if that's the case. Do patients with Dravet syndrome, which have a sodium channel mutation, do they have aberrant neurogenesis, right? Is this something that we can generalize to other epilepsies? And I think that would be for
and
the.
You imagine you're going to be doing this in organoids. I mean, this is exciting. I mean, what are the sort of disease in a dish models? Because I don't think in terms of that just yet. I'm learning. So can you kind of connect
so
that?
The one way that in Jenny's lab that this neurologist kind of started going down was modeling patient diseases, right? So he had access to patients, and we could collect their blood essentially, and then eventually with Chris's help, at the stem cell core, start reprogramming the patient blood samples to their iPS cells, and then we can model their diseases with organoids, with 2D models, with whatever, to kind of just understand the genetics of, I think, epilepsy. We're starting to really, in the last 10, 15 years, really started understanding that the genetics is complicated and difficult, and there's a lot more we don't know than we do know. And so everybody's starting to pick, genes that are involved, and we're starting to really understand that certain epilepsies were classified a certain way, but it's even further diverse because even within that specific classification, there's different mutations that could do different things. So it's this approach really helps kind of facilitate that. I think there's a hurdle when it comes to modeling in a mouse, just in terms of generating the mouse, studying in it, relating it back to the patient condition, patient mutation. So using the iPS cell model approach, it's a little bit higher throughput. It's a little bit more, you can retain a lot of the patient background information, study some of that.
Yeah,
I think one of the things, oh, go ahead, Jenny.
well, I was saying that I think I've always had this profound interest in translational neuroscience. And so, when we started working on adult neurogenesis and its relationship to epilepsy using the mouse models, it became very clear to all of us immediately that it's important to dissect the mechanisms, but the vast majority of patients already come to the clinic with epilepsy. So, although we don't really know how elucidation of the adult neurogenesis mechanisms, they still may help, or they may help that, that population of patients. We just don't know. But at the same time, the organoid models allow us to kind of go back in time, in a sense, and recreate the process, right? I mean, basically human tissue is inaccessible, particularly pediatric tissue. And so we just don't know if one of the reasons why a lot of brain disorders, if there is a developmental origin. So that's also an open question. So I think, one of the things that really benefited us before was, I think just establishing my lab at a medical center where we were able to, collaborate with neurologists and really be able to try to, bridge that gap between the basic to the clinical side. And that's something, of course, that we've continued to do here at UTSA. And then we've established the stem cell core to try to facilitate that.
And I think one of the interesting things that you can do is, with the mouse, you're also just getting a snapshot, right? You pick a time point and you're seeing what's going on at that exact time point, where with Jenny's lab, they've been doing a number of living studies. And so you can go in and see what's happening in those organoids before you start the experiment, and then follow it down the road too. So you can start to get at that question of what was going on in those stem cells before you started the experiment. And maybe that can explain some of the variability you're seeing after the insults. So I think they're really complementary techniques to try and break down what's happening.
Really exciting. Well, thank you guys for talking about this. Do you want to, we can call it, are we at 35 minutes? I think we are. But there's so much we could talk about. I mean, Chris, with a lot of the dopamine stuff that you're doing, so exciting. We'll have to come back and talk to you about that. And Zane,
Sure.
Best of luck to you. We hope to see you in a couple of years with some new data and a new podcast. So
Thank you. That'd be great.
Looking forward to that. Thank you, Jenny. Thank you, everyone. This has been Neuroscientists Talk Shop.
Thank
Thank
you.
you.
Thanks.